Good evening. Good evening, all of you. Okay, Hasandi. Get well soon. Right. Turn on your videos, all of you. Hasandi, okay. Hari, last time actually we were unable to do the session, especially because here the things were okay. Last year, actually, it was something like a trial. You can see that the board is there. I'm here. That's only, but here there is a big setup. Now, earlier, you can remember that here there was a large light bowl, like it was glittering here, so you were not able to see the things properly. But now it is okay, right? Then on the pain on. So I have to make the setup for that, right? Okay, this ini. The methanol lighting, Udinui on sides, everything. So I had to get the support of a resource person. So when he is coming, I can't say that I have the class, so you can't do it. Kiela anything to do the classes properly in the coming year. I have to be prepared, right? Any side. Last time I couldn't do actually. So I will just revise the things we had done on the first day, right? Then we can continue. So we discussed about, we started to discuss about the major biomolecules, right? So there are some more to turn on the videos. Could you please turn them on with, I don't need to mention the names now. Yes, there are some more. Hari. So major biological molecules, first we mentioned about the elements, the four most abundant elements in living matter, OCHN, right? We mentioned about them. Those who have joined today itself, as well as those who were absent on the 2nd of December, I have shared the recording with all of them. So you need to watch that, right? You have to watch it before today's session. If you are unable to do so, you have to watch it at least later. Then only you will find the flow of the lesson, right? Then chemical compounds of life, we divided them as organic and inorganic. Under the organic compounds, we mentioned about carbohydrates, proteins, lipids, and nucleic acids, which are considered to be the four major biomolecules. And we mentioned even the vitamins come under organic compounds. And under the inorganic compounds, we mentioned about water, minerals, and gases. And we mentioned about the exceptions as well. Then we started to discuss about carbohydrates. We gave examples for carbohydrates, and we mentioned the elemental composition of carbohydrates as CHO. And we mentioned that the common molecular formula of a carbohydrate is CX within brackets H2OY. So we gave examples for this. Then we classified carbohydrates as sugar and polysaccharides. Then the sugar we divided into two categories once again, monosaccharides and disaccharides. So we explained about them, the characteristics of sugar and polysaccharides, and how do they differ in their chemical composition as well. ටික අපි කතා කළා නේද? Classification of carbohydrates. We did it here. මතකද? Those who are there, you can remember, right? Hari. Then we today, I think we have to continue from this, right? Monosaccharides, disaccharides, and polysaccharides, their location as well as their importance, other important facts, right? So here we have taken the monosaccharide. So I have taken this from your textbook itself. So, and on the first day I told actually most of the questions, almost all the questions are prepared based on your textbook, right? So In addition to the teacher's guide, this also acts as a guide. So they ask questions based on this. And if you have got the textbook, now you may have seen that there are certain things named as extra knowledge. So from those things, actually, they don't make questions for the O-level paper. But sometimes for the term tests, they will make questions based on them. ඒ නිසා ඒ ටිකක් දැනගෙන ඉන්න. ඕනෙම දේවල් ටික we are all focusing on the O-level exam, right? Therefore, we will be preparing you all for the exam, O-level examination. Hari. You have to remember them carefully. These things you have to definitely remember. That's what I wanted to say since I have taken these from your textbook. Glucose, fructose and galactose. These are the three hexoses or sugar that contain 6 carbon atoms that we mentioned as examples. Glucose, fructose and galactose for monosaccharides. If we take glucose, it is available in ripened fruits. In ripened fruits, there is a sweet taste, right? That is because of the presence of this glucose, sugar. And in addition to glucose, there is something else as well we will mention. Fructose is also available. Fructose is known as the fruit sugar. Honey. Then in bee honey also we find glucose. So you learn that polysaccharides, starch, glycogen, glycogen, cellulose. In all these three, the basic unit of structure is glucose, right? Monomer of cellulose, starch, galactose. In all these, cellulose, starch and glycogen. These three are polysaccharides. In all these three, the basic unit is glucose, right? The end product during hydrolysis, of all starchy food is glucose. During hydrolysis, they will be converted back into polysaccharides, into disaccharides, then into monosaccharides, hydrolysis. So this glucose is the end product of digestion of these carbohydrates, right? Then this simple product, this monomer will be absorbed into bloodstream. Then this undergoes cellular respiration. This glucose reacts with oxygen producing energy. That is what we call as cellular respiration. Plants produce glucose during photosynthesis. Glucose is the main product of photosynthesis. In grade 11, second unit, we are discussing about photosynthesis. So glucose is the main product of photosynthesis. Then energy is released during breaking down of glucose in cellular respiration. So glucose reacts with oxygen forming energy during cellular respiration. Now, actually, later we will discuss about photosynthesis as well as cellular respiration. Both, right? Glucose is absorbed into blood. This is the end product of hydrolysis. That is glucose. These are important, right? Nang apita then grade 11 wala igena gannakam we can just write them and keep during photosynthesis. I will find a space. You also have space here, right? නැත්තම් here. මෙතන ඉඩ තියෙනවා නේද? උඩින් එකක් ලියලා යටින් තව එකක් ලියන්න පුළුවන් පහලින්. නේද? So during photosynthesis, අපි ලියලා පෙන්නමු photosynthesis. මොකද වෙන්නේ? Carbon dioxide. And we will write the word equations. Carbon dioxide plus water react in the presence of light, energy and chlorophyll. producing glucose and oxygen. Glucose plus oxygen, producing glucose and oxygen. This is photosynthesis reaction. Um, the word equation, carbon dioxide plus water in the presence of light and chlorophyll, it produces glucose and oxygen. So glucose is the main product of photosynthesis, right? Then in cellular respiration, actually, they make use of this glucose during cellular respiration. Cellular respiration. මතක තියාගන්න ලේසියට මතක තියාගත්ත හැකි. It is the opposite of photosynthesis කියලා. It is something like the opposite of photosynthesis. So here you can say glucose reacts with oxygen. Right here you can see, right? Glucose reacts with oxygen, forming. And we know exhale there contains carbon dioxide because carbon dioxide is also a product of cellular respiration. Carbon dioxide, water vapor, and also energy. Energy is the main product of cellular respiration. Cellular respiration is done in order to obtain energy. Here glucose is the main product. Here energy is the main product. So carbon dioxide and water vapor are byproducts of cellular respiration. Yes, Jatu Sharan, can you turn on the video? Dihen. Manusha, Yashan, Vinudi. Yes, done. හරි නේද? ඔන්න මතක තියාගන්න grade 11 වල lesson එක කරනකම් you can remember. හරි. Then about fructose. Yes, fructose is available in ripened fruits. Therefore, this is also known as the fruit sugar. These can come for MCQs, right? Fructose is the fruit sugar. This is available in ripened fruits, bee honey, pumpkin. They have a sweet taste, right? Carrots. The sweet taste is there because of the availability of fructose. Fructose is formed during ripening of fruits. Raw fruits, they don't have a sweet taste, right? They basically have starch. Right? Then when they become ripen, due to enzyme activity, this starch is gradually converted into simple sugar. Maybe it can be එතකොට disaccharide එකක් තිබුණොත් if it is sucrose, it is converted into glucose and fructose. Likewise, they are converted into simple sugar on ripening. That is why they get a sweet taste. Then that this is the sweetest sugar. Fructose is considered to be the sweetest sugar, right? Sweetest. Then galactose. Galactose is available in dairy products, milk-based products, right? The end product during hydrolysis of lactose. Lactose on hydrolysis, it forms glucose and galactose, right? Matakai neda? Glucose and galactose. The end product during hydrolysis of lactose. This does not have a sweet taste. Ogolo fresh milk. Bila තියනවා නම් දන්නව නේද? If you have tasted fresh milk without adding any flavoring agents, දැන් අර තියෙන vanilla flavor, strawberry flavor, chocolate flavor, එහෙම මොකුත් flavors නැතුව, you know, the fresh milk. If you taste some fresh milk, you know, there is no any sweet taste there. That is because of galactose. Galactose does not have any sweet taste, right? So galactose is available in dairy products. Hari. That is all about monosaccharides. Then we'll move on to disaccharides. Di means two කියන එක නේද? We learnt. So two monosaccharides combine in order to form a disaccharide. Right? One of the disaccharides you are given maltose. Right? Maltose. Maltose is available in germinating seeds. Then actually these seeds contain starch at the beginning. Right? Then when we are germinating the seeds, we soak the seeds in water. In grade 6, when you are germinating green gram, you had to soak them in water for one night. So when we soak them in water, the enzymes inside those seeds become active. Right? These enzymes convert that starch into maltose. Right? Starch, maltose, then maltose is then converted into glucose, simple sugar. Maltose is available in germinating seeds. Union of two glucose molecules form a maltose molecule. Glucose plus glucose, maltose plus water, removal of water. මතකයි නේද අපි කතා කළා? What do you call this reaction? A water molecule is removed. What was the name given? Condensation, removal of water. Good. So this is an intermediate product of starch hydrolysis. Starch hydrolysis starch hydrolysis. In the starch hydrolysis, what happens? Starch is first converted into maltose, and then maltose is converted into glucose plus glucose is formed by maltose. And a maltose on hydrolysis, it will be converted into glucose. It says it is an intermediate product of starch hydrolysis. Madding in a maltose in starch hydrolysis. Did you get that? Lazy to answer the questions given. Then second disaccharide, that is sucrose. Sucrose. So that is available in, it is available in white and brown sugar. The sugar we consume. Sugar cane and beet. Sugar cane, the sap contains sucrose, and also beet. Beetroot has a sweet taste. Now, pumpkin and carrot fructose වලට. Now here for beetroot we say that it contains sucrose. Sorry, some fruits, white and brown sugar, sugar cane and beet. Some fruits contain sucrose. and phloem sap in trees. ඒ ගැනත් අපි කියනවා in grade 11 when we learn about photosynthesis. For the moment, so the main product of photosynthesis is glucose. Then it is temporarily stored in leaves in the form of starch. The leaves වල store කරන් ඉඳලා බෑනේ. It should be translocated to the other parts of the plant body for storage and consumption, like fruits, maybe the stem, the roots, to those parts that should be translocated. So food translocation is done by the phloem tissue. You have learned about it in grade 7, නේද? Phloem tissue. In phloem, how this food gets translocated? It gets translocated in the form of a sucrose solution. Sucrose solution එකක් විදියට තමයි phloem එක දිගේ යන්නේ. Right? That is why here it says phloem sap in trees. So union of a glucose molecule with a fructose molecule or fructose molecule forms a sucrose molecule. Glucose plus fructose, condensation, removal of water, it forms sucrose. Right? We learned about this reaction last time. Harida, and if you have any problem, you have to ask, right? Come අහගෙන ඉන්න බෑ. දැන් අපි ජනවාරි වල පටන් ගමු questions discuss කරන්න. lesson එකක් කරලා තියෙන්නේ බෑ questions වලට යන්න. ඊට පස්සේ we are moving on to the second unit from which you are getting questions definitely for the O level paper. ඒ කියන්නේ මේ වැනුත් එනවා. ඒකෙන් ලොකු ප්‍රශ්නයක් අනිවාර්යයෙන්ම එනවා ගොඩක් වෙලාවට. Right? හරි. Then the last one, the last disaccharide, that is lactose. Right? Lactose. Lactose is available in dairy products. දැන් අපි ඉස්සෙල්ලා galactose කිව්වේ as the monosaccharide. Galactose plus glucose forms lactose and water. Removal of water. ඒ remove කරපු water molecule එක තමයි මේ පැත්තේ ලියන්නේ. Union of a glucose molecule with a galactose molecule forms lactose. This is the only sugar that is absent in plants. දැන් අපි කියවනේ දැන් galactose did not have a sweet taste, right? Galactose does not have a sweet taste. Here lactose should be slightly sweet because it is the combination of galactose and glucose. Since glucose is sweet, it forms lactose and water. And remember, this is the only sugar that is not available in plants, right? This is not sweet as sucrose. ඒ වගේ පැණි රස වෙන්න බෑනේ. Sucrose was due to the hydrolysis of glucose and fructose. Here, glucose and galactose. Galactose did not have any sweet taste. ඒ නිසා අර වගේ පැණි රස නැහැ. Sorry. The percentage of lactose in cow's milk, according to the composition, is about 4 to 6%. The percentage of lactose in human milk is about 6 to 7%. That is bit higher than cow's milk. And not only that, there are so many other differences. That is why the small babies are said to be fed upon mother's milk only. It contains lot of nutrients, as well as it will make a good combination in between, close relationship in between the mother and the baby. Okay? Hari. So here you are shown මේ වඳින්න පාඩම් කරන්න ඕනේ නැහැ. අම්මෝ බය වෙනවා දැන් මේවත් අඳින්න වෙයිද කියලා. නෑ නෑ. ඒක දුන්නේ නිකන් පෙන්වන්න විතරයි. හරිද? To show how this bond is formed. ඔන්න දැන් ටික දෙනෙක් disappear වෙලා ආපහු. හරි අමාරුයිනේ appear වෙලා තියාගන්න. මොකුත් zoom එකේ නෑනේ option එකක් එහෙම. video off කරන්න දෙන්න බෑ එහෙම කියලා. මොනවා නම් හොඳයි නේද? වෙලාවකින් disappear වෙනවා. දාසයි ඉතින් ලස්සන මූණු බලාගෙන. Yes, Hiruja. මොකද Hiruja සැරින් සැරේ disappear වෙන්නේ. හරි, තව ඉන්නවා. Turn on the videos. Manusha, Manusha. Yes, Venuka. Hari. එතකොට පොතේ මෙන්න මෙතන පෙන්නනවා how the water molecule is removed කියලා. මේ these are two glucose molecules. This H2O නේ water කියන්නේ hydrogen from this molecule and OH from this molecule, and that H2O is removed and the glycosidic bond is made in between the two glucose molecules. ඒක දුන්නේ අර ටිකක් එහාට හිතන අයට. සමහර හිතනවා දැන් එක එක අය ආස දේවල් තියෙනවානේ. සමහර අය මේවට ආස ඉගෙන ගන්න. කොට හිතනවා කොහොමද මේක උනේ? අන්න ඒගොල්ලන්ට උත්තරයක් විදිහට තමයි I just added this uh, structural formula, right? Structural formula and the reaction. ඒක මතක තියාගන්න ඕනේ ඔයගොල්ලෝ. හරි. ඊළඟට ඔන්න තව වචනයක් එනවා. Reducing sugars and non-reducing sugars කියලා. Reducing sugars, once we say reducing sugars, on another thing to remember, when we say reducing sugars, it includes all monosaccharides. These are reducing sugars. When we say non-reducing sugars, අපි ඔයගොල්ලන්ට grade 10 වලට මේ මතක තියාගත්තාම ඇති, sucrose is the most common non-reducing sugar. දැන් මෙතන තියෙන reducing agents කියලා ඔයගොල්ලෝ එච්චර දුරට හිතන්න යන්න ඕනේ නැහැ. මෙතන මතක තියාගන්න all monosaccharides are considered to be reducing sugars, and sucrose is the only non-reducing sugar that we are discussing. මොකද ওই වචන දෙක අහනවා සමහර වෙලාවට. Next we are going to discuss about the tests to identify carbohydrates. එතකොට සමහර තැන්වල කියනවා test වලදී this is for reducing sugars and this is for non-reducing sugars කියලා. එතකොට ඔයගොල්ලන්ට පැටලෙන නිසායි මම මෙතන කියලා තියෙන්නේ ඒක. Right. Polysaccharides. So we mentioned about cellulose, starch and glycogen. So if we take cellulose, cellulose is essentially available in the plant cell wall, right? Cell wall of plant cells. Ilangada, the fiber, some plants contain the, in all the plants, the fiber are there. Grade 11 first unit, we are learning about the plant tissues. There we mention about this plant fiber, that is a type of cells known as sclerenchyma. Hari. So remember cell wall of plant cells. Then api kiyana fiber. Some plant materials contain more fiber. Then this fiber means cellulose, right? So remember this cellulose does not get digested in our digestive system. Abai api kiyana in a balanced diet we have to obtain all the nutrients with vitamins, minerals, and also fiber should be there කියලා. නේද? Fiber facilitates the digestion, right? It facilitates the removal of undigested materials. Right? The removal of fecal matter becomes convenient. Then the person is protected from cancer-like conditions in the intestine, especially the large intestine. Hari. Because it does not get digested in our digestive system. Kendi sahita ahara can fibrous food. So they facilitate the removal of the undigested materials, what we call as the fecal matter. And when we consume food with lot of fiber, that means fruits, vegetables, grains, especially fruits and vegetables, and also green leaves. When we consume those, actually it helps to control the blood glucose level as well, because normally we have to take the salads before the main meal, right? අපි dessert කියලා සමහර වෙලාවට පස්සෙත් ගන්න. හැබැයි actually we have to take them before the main meal. So when we consume fruits and vegetables, we feel that our tummy is full, so we don't need to eat much. එතකොට we minimize the starchy food, නේද? Then that means starch finally it is converted into sugar, no? එතකොට that is good for people who are suffering from diabetes and as well as who need to control the blood glucose level. It is good to consume more fibrous food. හැමෝටම හොඳයි. If you eat only rice and meat, then you don't get enough fiber. Rice has a little bit, but you have to essentially consume at least 5 fruits or vegetables in your diet. හරිද? I think it is not digested in the human digestive system, but it helps to avoid constipation. That means in constipation, it is difficult to remove the fecal matter. They become uh, solidified, more solidified. Therefore, it is difficult to remove fecal matter. Then this is known as constipation. Though it is not digested in our digestive system, it gets digested in the case of cow, the termites. ඒගොල්ලෝ එහෙම අර කඩදාසි wood කන්නේ ඒක නිසා තමයි. That gets digested in them. Then starch, grains, yams, jack, breadfruit. bread උත් හරි. මෙතන නම් දීලා තියෙන breadfruit කියන එක, right? Breadfruit, sweet potato, right? Manioc, yams, ඊළඟට potato, they contain starch. The type of carbohydrate that is stored in plants is starch. Very important. The starch should be stored in a particular form in plants and animal bodies. So, the stored type of carbohydrate that stores in plants is starch. Right? So, in plants, carbohydrates are stored in the form of starch. Very important. Then glycogen. Glycogen is available in animal liver as well as the muscles. That happens once again in grade 11, 6th unit we are discussing. How the blood glucose level is maintained. If there is excessive amount of glucose in our blood, that is converted into glycogen by insulin. Right? So insulin converts excess glucose into glycogen and it stores them inside our liver as well as the muscles. And glycogen is the stored type of carbohydrate in animals, right? In animal bodies, carbohydrate is stored in the form of glycogen. In plants, it is in the form of starch. Remember. සමහර අය පටලවා ගන්නවා ටික දවසක් යනකොට කියනවා this is cellulose කියලා. නෑ. Stored type of carbohydrate in plants is starch. Stored type of carbohydrate in animals is glycogen. හොඳට highlight කරලා හිතෙත් highlight කරගන්න. Right? Starch and glycogen. Hari. Any problem? And then දැන් හිතනවා ඇති හරි ලේසි 10 වසරේ වැඩ මේ ලියන්න දෙයක්වත් නැහැ කියලා. මේ ටිකේ තාම වැඩි ලියන්න දුන්නේ නැහැ. ඊළඟට හොඳට ලියන්න හම්බ වෙනවා. ඉස්සරහ පාඩම් වලදී. දැන් පාඩම් කරන් තමයි තියෙන්නේ මේ ටිකේ. මේවා ආයි තියාගන්න එපා ඉස්සරහට. ඔයගොල්ලන්ට දැන් grade 9 third term test තියෙන්නත් පුළුවන් නොතියෙන්නත් පුළුවන්. But you have to be prepared for the coming term tests as well as your ultimate objective is to face the O-level exam properly. ඉතින් ඒ නිසා දැම්ම පාඩම් කරන්න. මම කියවනේ weight එක කොහොමද කියලා. නේද? In grade 10 there are 20 lessons. In grade 11 there are altogether 15 lessons. Then that means 35 lessons are there for O-levels. ඉතින් ලේසි නෑ. ඕවා එක දවසෙන් පාඩම් කරන්න බෑ කොහොමත්. දැන් ඉඳන්ම පාඩම් කරලා short notes හදනවා නම් හදලා නම් පාඩම් කරන්න. You have to make them by now, not later. මම හදලා තියාගන්න. එතකොට grade 10 first term test will be held before April. So for that you have to be prepared with not 5, 6 lessons. 6 lessons are there for grade 10 first term test. හරිද? ඒ නිසා දැම්මම පාඩම් කරගන්න while enjoying your holidays. Right. Significance of carbohydrates. How carbohydrates become important. As an energy source. Carbohydrates act as a primary and major source of energy for the functioning of cells. අපි ඉස්සෙල්ලා cellular respiration reaction එක ලිව්වා නේද? We wrote the reaction for cellular respiration. So there you saw that glucose glucose reacts with oxygen forming energy, right? So here it says glucose is produced by the hydrolysis of complex carbohydrates. Then this glucose react with oxygen forming energy during cellular respiration. As an energy source, carbohydrates become important. ඔන්න මෙතන ආයි දාගන්න cellular respiration කියලා. Cellular respiration. අපි කලින් ලිව්වා equation එක. Then as a storage compound, in the table, as the last point we mentioned, in plants it is stored in the form of, yes, in plants it was stored in the form of starch, and in animals it was stored in the form of glycogen, storage compound. Hari. Then third one, as a structural component in plant cell wall. In plant cell wall, කවුද? That is cellulose නේද? Cellulose is available in the plant cell wall. In each and every plant cell, in the wall we find cellulose. Hari. Then as a constituent of nucleic acids, now as the fourth major biomolecule, we are going to discuss about nucleic acids. Till then, we will mention there are 5 carbon monosaccharides available in once we say nucleic acids, there are 2, DNA and RNA, right? So in DNA, we find deoxyribose sugar. In RNA, we find ribose. හරිද? ඔන්න ඒකටත් examples ලියමු. දැන් මේක ගැන worry වෙන්න එපා. We are discussing about nucleic acids in detail in this lesson. Example, අපි nucleic acids කිව්වහම DNA, RNA දෙකම තියෙනවානේ. DNA and RNA. In DNA, we find deoxyribose sugar. Deoxyribose sugar in DNA. In RNA, we find ribose sugar. Ribose sugar. මේ දෙන්නම 5 carbon sugar, right? ඒ නිසා අපි ඒවට කියනවා pentoses කියලා. 5 carbon sugar. Therefore, we call them as pentoses. Glucose, fructose, galactose, these are 6 carbon sugar. Therefore, we call them as hexoses. 6 carbon sugar. ඒවත් additional information, right? DNA deoxyribose sugar, and in RNA we find ribose sugar. Yes, පුතේ. සෙවිඳු, තිහෙන්, මනුෂ, යශාන්, තිහන්සා, Hari. Then as the fifth function we'll say, it acts as a transportation compound in plants. Number 5, as a transportation compound in plants. What is the example, Miheli? As the transportation compound in plants, what is the example? මොකද්ද පුතේ? Sucrose. Sucrose is the example. Sucrose. ඉහේලි වයි විහංගිව හොයාගන්න අමාරුයි. මොකද තාම අක්කාගේ නමින් තමයි සේව් වෙලා තියෙන්නේ. සොනාදි කියලා තමයි තියෙන්නේ. So ගොඩක් වෙලාවට මට පැටලෙනවා. රීනේම් කරගන්න ඕනේ නේද? හරි. Then තව එකක් දාමු අපි sixth importance. Provide dietary fiber. Provide dietary fiber. Example. Yes, Dihen, what is the example? Dietary fiber. Yes, Dihen, we mentioned. Oh, what is dietary fiber? What's the example? Venuka? Venuka? Oh, Venuka gihilla koyada. Yes, Venuji. Cellulose. Cellulose. Provide dietary fiber. Example, cellulose. Oh, the other ඉස්කෝලේ ගියාට marks හම්බුණාද? Grade 9. Those who had the term test, just send me the marks if you received. Hari. Okay, Hiruja, well done. 95. අනකම් ඉන්නානේ. හරි. එවන්න බලන්න. marks ගත්ත අය. හිරුජයට විතරද marks හම්බුනේ? Right. Then we are going to discuss about the tests to identify carbohydrates. Actually, some of them you have discussed earlier, neda? Hari, number 1. That is, I will share the videos for these, right? After discussing, I will share. And the first one is the starch test. Starch test. Number 1. So you have done this. So there you can take the food item directly and you can add a few drops of the iodine solution, right? And you can observe the color change. දැන් මෙතන ඔයාලගේ textbook එකේ කියලා තියෙන්නේ food extract එකක් ගන්න කියලා. You have to grind the food and mix it with water. Then only you are going to add few drops of iodine, right? So what is the color of iodine solution? Color of iodine? Oh, කොයිද? Yes. No, nepute. Before adding, what is the color of iodine solution? That is yellowish brown. Yes, iodine is yellowish brown. Then when we add it to a particular food item, here you can see slices of potato. When we have added this iodine solution onto slices of potato, you can see that the color you mentioned, dark blue, නැත්නම් purplish blue කියලා කියනවා. Blackish blue කියලා කියනවා. ඒ කළු පාටට හුරු නිල් පාටක් වගේ තමයි පේන්නේ. Right? හරි. එතකොට අපිට ඔන්න මෙතන මම ලියන්නම් the color of iodine, that is yellowish brown. Yellowish brown. we'll write this method here. Yellowish brown is converted into අපි පාට ටික ඔක්කොම කියමු. dark purple කියන්න පුළුවන්, purplish blue කියන්න පුළුවන්. purplish blue, blackish blue කියනවා, dark purple, right? ඔන්න වගේ. Oh, හරි. So here we'll write the steps. We are going to obtain the extract, right? Food extract. Take small amount of food and mix with water. Then you have to add the iodine solution. Then this is the observation, expected observation. Add a drop of iodine solution. Then you will be able to see this color. හරිද? Making එක පෙන්නලා ඉන්නම්කෝ. I will show you this activity which you have seen earlier also. I will show you something related to carbohydrates, and after that we'll watch the starch test.
Carbohydrates, everything you need to know. Carbohydrates, or carbs, are the primary energy source for and almost all other living organisms. They are one of the three nutrients in the body. They are polysaccharides made up of many saccharides bonded together in large complex structures. Carbs occur as starches, sugars, fiber that we get from plant food and dairy products. While their byproducts have other uses, typically take in carbohydrates to generate energy from them. Respiration. A single gram of carbs generates up to 4 calories of energy. The body gets them by breaking carbohydrates. Let's look at why carbohydrates are so important. Uses of carbohydrates. The body uses carbohydrates for 1. Producing energy required to carbohydrates are the primary source of energy. That's an extra protein energy. 2. Storing energy that may later on. Energy is stored in the form of glycogen, mostly present in liver development of micromolecules such as RNA, DNA, and energy of the body. Strong stress levels. 5. Make sure that fat proteins not used up for and stay stored in the body. Health benefits. The health benefits for carbohydrates. 1. They provide ample fuel, nervous system, heart muscle. 2. Good gut health because of diet. Fiber is digestible in you, but it is critical to help push food along, help you feel full, and keep away from 4. All in check. Liver. Healthy carbohydrate. Make sure this happens. The daily recommended amount of carbohydrate, 1,000 calorie diet for an adult, hundreds of grams. However, also need to be kind of carbohydrate. While you cannot digest dietary, there are no actual restrictions. However, at least grams of it a day. What you need to limit is excess sugar can cause severe health problems, such as obesity, diabetes. To limit sugar as much as possible, you should avoid taking more than 50 grams or 10% of your daily intake a day. Risks: excessive carbohydrate can lead such as type 2 diabetes, excess blood sugar can result in excess carbohydrates are stored in the body will cause you to gain weight. Heart disease. Gaining weight puts strain on your heart as blood vessels become narrowed due to more fat in the body. High cholesterol. Fat content. A high amount of cholesterol. Thank you for watching our video. Please do not forget.
Right. Then I will share the starch test, right?
An important concept. An important constituent of our daily carbohydrates consumed in the form of starch. Starch is a high calorie food that converts to sugar digestion. Starch is also produced by all green plants as an energy store. Today we will experiment to test the presence of starch in potatoes, rice, sugar, salt. For this, iodine solution, yellow brown in color. Note the change in the color of iodine, rice, and potato. Don't turn the iodine back. Put some iodine on the tube. The color does not change. Salt too does not change color. This proves that sugar and salt do not contain starch. While the starch in rice and potatoes reacts with iodine to form a blue compound. So the reaction is because of the black color, the formation of polyiodide, the reaction starch iodine.
Okay. Hari. Then we'll see the second one, that is the test for reducing sugars or monosaccharides. දැන් මං කියවන all monosaccharides are considered to be reducing sugars කියලා නේද? අපි ඒකට කියමු test for reducing sugars කියලා. All monosaccharides and some disaccharides are included in this test for reducing sugar. Within brackets, I will say all monosaccharides. And some disaccharides. right? Hari. So here what you have to do is on observation එක ඉස්සෙල්ලාම දීලා තියෙනවා. Here what you have to do is you have to take a solution of glucose. You can take a glucose solution into a test tube by dissolving some glucose in water, right? Then you have to add few drops of Benedict solution which is available in the laboratory, right? අපි මේ පැත්තෙන් අඳින්න පටන් ගමු because the observations are given here. You take 2 milliliters of glucose solution, right? We'll say 2 cubic centimeters of glucose solution, right? To that, you have to add 2 cubic centimeters of Benedict solution, right? Benedict solution. නැත්නම් Benedict reagent කියලාත් කියනවා. After that, you have to shake this well, shake the two components well to mix them together. Immerse this boiling tube or the test tube in a water bath. Water bath and heat it up. අන්න ඊළඟ step එක මම මෙහෙම අඳින්නම්. You have to keep this inside a water bath. And I have kept a beaker on a tripod and this boiling tube or the test tube is inside. Now the mixture is there, right? The mixture is there. You have immersed this in a water bath. Here there is water and you have to apply heat it up. අපි steps ටික මෙතනින් ලියමු. Then you will be able to see those color changes. මොනවාද? First you will see actually Benedict reagent is blue color, right? Therefore you will see the blue color first. This is number 1. Then it becomes green, right? Then it becomes green yellow වගේ එකක්. Yellow මත් නෙවෙයි, in between green and yellow. We'll say green yellow. Green yellow. තව ටිකක් එහායින් පටන් ගන්න වෙනවා නේ මට. කියන්න. Blue, green, then green yellow, then orange, and finally it becomes a brick red precipitate. අර ගඩොල් කුඩු වගේ brick red කියන්නේ එකයි. ගඩොල් වල තියෙන අමුතු රතු පාටක්. අන්නේ විදිහේ precipitate එකක් එනවා. It deposits on the bottom. Brick red precipitate. මේක උඩ ලියන්න ඕන. You have to manage your space properly. Brick red precipitate. ඔන්න මම පැත්තෙන් ලියනවා precipitate කියන වචනේ ආපහු. Precipitate. Brick red precipitate is obtained. ඊට පස්සේ අපි මෙතන පහලින් steps ටික ලියමු. I will read out. You can write down the steps. මේ ටික දාගන්නකෝ. ඔන්න ඕගොල්ලෝ තමයි පාඩම් කරන්නේ. ඒ නිසා ඔයාලගේ note එක ඕගොල්ලන්ට පැහැදිලි වෙන විදිහට ලියාගන්න. Yes, Akitya. Inudi. Manusha, where are you? And what happened to Venuka? Yes, Denuli. Done. Hari. ඔයාලා නද ඉස්කෝලේ ගියාය? Just raise your hands, those who went to school today. Okay, most of you have gone. 20. Amika, you didn't go. School didn't start.
What teacher we have holiday?
Okay. Then Kobalan, those who are going to have the grade 9 third term test, going to have, some have finished already, no? Those who are going to have the grade 10 third term, sorry, grade 9 third term test. Devmini, Ashan, you are having, right? ඉදසල් තියනවා නේද? හරි. yes මට මතකයි. Miheli, you are going to have. No, you don't have. Manushika, I think Devmini, Manushika, you are from this Yashin, from the same school. Am I correct? Oh, ne? Hari. Savindu, you have. You are going to have. Test තියලා ඉවර වෙච්ච අයත් ඉන්නවා අනිත් අයට නෑ කිව්වා නේද? Actually, it may be a great relief for some of the students, like those who have lost their notes and all, ne? Because of the adverse weather, some students have missed their notes and everything. එනිසා ඒගොල්ලන්ට ඒක ලොකු relief එකක් වෙනවා නේ. Blue, green, green, yellow, orange, and the brick red precipitate. Hari, have you all finished? Hari ne, shall we write the steps? එමුද? මෙහෙම ලියන්න. First one, take 2 cubic centimeters of glucose solution. Take 2 cubic centimeters of glucose solution into a test tube. Into a test tube. Then next, take 2 cubic centimeters of glucose solution into a test tube. Next one, add a similar volume of Benedict solution. Add a similar volume of Benedict solution. Then next one, immerse the about test tube. Immerse the about test tube in a water bath. In a water bath. In a water bath and heat. Alright, those are the steps. So I will share that test. I will share the video. Just have a look at this. And
Hi, I'm Martin Asher from Berry College, and I'm a biology teacher, and today we're going to be doing a simple food test, determining the amount of glucose or sugars that's present in some of the foods you would normally eat for breakfast. So we've sort of already set up an arrangement of you might eat for breakfast. Got rice, oats, beans, biscuits, and frosties. And what we have in front of each one is the amount of sugar that is present per 100 grams. So if you were to have something like rice for your breakfast, in terms of low sugar content, zero, then all the way to the other end of the scale, frosties, which has 37 grams of sugar per 100 grams, and very, very high sugar content. So what we're going to do today is we're going to do a genetics reducing sugars test, just a reagent test to detect the colour change, how much glucose is present in different foods or different solutions. So normally, if we were to do this in class, we'd be taking the amount of sugars present and we would dissolve them into a large amount of water. You would have unknown concentrations and then you would determine how much sugar is present and its known values what we're going to be looking at shortly. So what we have three different solutions. We have a glucose solution, a water solution, and then we also have a well-known energy drink. See how much sugar is present in it. We're going to put 5 centimeters cubed into each test tube here, as well as the Benedict's reagent, which is what's going to produce that colour change, and then we can compare to known values how much sugar is present.
this is Benedict's reagent, right? It has the color of copper sulfate, right? Actually, it contains copper sulfate as well.
Each one in water, which is 80 to 100 when rolling to the make sure it's hot. Wait.
So you can see the color change gradually.
Choose salt, water, use sugar presence, so there's no actual choose your favorite syllabus, with a concentration of the choose on you a well think will have a concentration also on in color record. What we call results, no results. Range of sugar ranging from 0 all the way to 8 and milligrams. And the lower orange color amount of sugar, which in this case already break. So we
Hari. So you saw the steps. Once you add the Benedict region and immerse it in a hot water bath, you can see the color changes, right? In the order blue, green, yellow, orange, and brick red precipitate. And remember to mention this order of colors, right? When you asked about the observations. පිළිවෙලින් ලියන්න මතක තියාගන්න. හරි. Then we will move on to the test for non-reducing sugar, right? Test for, actually there we are going to discuss about sucrose. That is the third one. I'm going to erase the board, right? Right? So you can put number 3 and say test for non-reducing sugar. Example, here we are taking starch, right? Hari. So, sorry, not starch. Extremely sorry, that should be sucrose, non-reducing sugar. Sucrose. Hari. So, in your textbook, they say they follow the same steps for sucrose as well. Then you were unable to get a result. There was no any change, right? That means you take sucrose solution at the similar volume of Benedict region and shake well. Then after that, you have to immerse this in a hot water bath. There is no change in color, right? Then you have to do a slight change in between the steps. එක දෙයක් කරන්න ඕනේ. ඔන්න එක දේ මොකක්ද කියලා ඔයගලන්ට තේරෙනවා. Now we learned that sucrose is a product of what? Yes, it is produced due to the condensation of, yes, glucose and fructose, right? When glucose and fructose combine, a water molecule is removed and sucrose is formed. So if we convert this sucrose into the reducing sugars, the simple sugar monosaccharides වලට convert කරගත්තොත් හරි නේද? එහෙනම් අපි you know that sucrose, due to hydrolysis of sucrose, අපි මෙතන water කියලා දාමු. Hydrolysis, then it will form glucose and fructose. Then these two will give a positive response for Benedict reagent. මේකට අපි කියුවේ මොකක්ද? Hydrolysis. Hydrolysis. එතකොට rather than adding water, we are going to add few drops of an acid. Acids provide මෙතන hydrolysis can actually hydrogen ions should be introduced to the medium. Therefore, we are adding a few drops of dilute sulfuric acid. හරිද? එතකොට sulfuric add කළාම මේකට ඔයගොල්ලෝ හිතා acidic medium එකක් වෙනවා. එහෙනම් to not to make the medium acidic, we can add a similar volume of sodium hydroxide as well. මේ steps ටික ඔයාලගේ textbook එකේ කියලා නැහැ. But when we watch the practical videos, they mention after adding few drops of sulfuric acid, you can add a similar volume of same concentrated sodium hydroxide as well, since it is a base. Hari, එච්චර දුර හිතන්න ඕනේ. Then we follow the same steps, and before we insert it in a hot water bath, we can add few drops of dilute sulfuric acid. Then it will be hydrolyzed like this, and glucose and fructose will give a positive result for the Benedict test. So the coloring observations blue, green, yellow, orange and brick red precipitate will be observed. හරිද ඔන්න මේක තමයි අපි දැන් කරන්නේ. සුක්‍රෝස් දෙන්නේ නැහැ positive result එකක්. නිකම්ම. එයාට hydrolyze කළේ නැත්තම්. For hydrolysis we have to add few drops of an acid. Right? හරි. එහෙනම් අපිට steps ටික මෙහෙම කියන්න පුළුවන්. අපි ඇඳලා පෙන්නමු නේද? ඉස්සෙල්ලා ලියුවානේ steps. 2 cubic centimeters of sucrose solution. Right? To this one you have to add 2 cubic centimeters of Benedict solution. ඊට පස්සේ at the same time you can add few drops of dilute sulfuric acid. Then after that you can keep this inside the water bath. Add number 1, 2 cubic centimeters of Benedict solution or reagent. Then few drops of අපි ඒක වෙන color එකකින් ලියමු. This is important, right? This is a difference that we do. Few drops of dilute sulfuric acid කියන එක මෙහෙම ලියනවා dil dot එකක් තියලා H2SO4. ඒ කියන්නේ dilute sulfuric acid. Right? Add few drops of dilute sulfuric acid. ඊට පස්සේ as usual immerse in a water bath and heat. දැන් මේ ටිකේ නිශ්ශබ්දව ඉන්න පුළුවන් වුණාට from January onwards, when we are starting the questions, you have to give me the answers, right? Brick red precipitate. Precipitate means it gets deposited on the bottom. In the activity, in the video, you saw that if we keep it for some time, this liquid substance gets deposited on the bottom of the test tube. Okay, Venuka, is it raining there? Raining at Mathur. Here also, I heard that it is slightly thundering, but no rain yet. And make it මොකද වෙන්නේ කියන එක තමයි I have mentioned at the bottom. Hydrolysis, when we add the acid, sucrose is converted into glucose and fructose. That means sucrose is hydrolyzed. You have to write the correct steps if you are asked to write the steps. Hari. If you have finished writing, you can have a little break. Just have a 2-minute break and come back. Drink some water and come back. ඔතන වාඩි වෙලාම ඉන්න එපා. චුට්ටක් නැගිටලා පොඩක් රිෆ්‍රෙෂ් වෙලා එන්න. හරි. ඔන්න වැඩිපුරත් වෙලාව දුන්න දැන්. To be refreshed, right? Okay. Now turn on the videos, all of you, for the rest of the hour. Hari. We are moving on to proteins. We have finished talking about carbohydrates. Now it's about proteins. So proteins also are essential for the for the growth of living organisms, right? Protein is an essential constituent in all living cells, right? So what are the elements present in proteins? The elemental composition. On carbon, hydrogen, oxygen, nitrogen is also available in proteins. CHON. These four are essentially available in proteins, and in addition to that, in some of the proteins we find sulfur as well in some of the proteins. එහෙනම් අපි කියමු carbon, hydrogen, CHON, oxygen, and nitrogen. CHON are always present in proteins, and sometimes sulfur can also be present. Sulfur can also be present in some of the proteins, right? An important point. 17% of mature human body is composed of proteins. 17% of mature human body consists of proteins, right? It is a complex molecule. It is then අපි polysaccharides ගැන කිව්වා. In the case of carbohydrates, they have undergone polymerization reaction කියලා. The monomers undergo polymerization and form the polymers. ඒ වගේ protein is a complex molecule made out of polymerized amino acid molecules. එතකොට protein එකක් හදන smallest unit එක තමයි amino acid. Right? The number of amino acids combine and form proteins. So we find 20 such amino acids. දැන් ඔයගොල්ලෝ මෙහෙම හිතන්න. Then there are how many letters in the English alphabet? 26 letters are there. So what are the patterns that you can write those 26 letters in different sequences? ඔයා මේ අකුරු ටික මාරු කර කර ලියනවා. කීයක් විතර ලියන්න පුළුවන් වෙයිද? අනේ A B C D පිළිවෙලින් ලියනවා. ඊට පස්සේ A B B ඊට පස්සේ මාරු කරලා ලියනවා. එහෙම එහෙම එකින් එක මාරු කර කර you can write a large number of combinations නේද? අන්න ඒ වගේ. There are 20 amino acids. Right? There are 20 amino acids. These amino acids combine in different sequences in order to form different proteins. Right? So, protein is a complex molecule made out of polymerized amino acid molecules. Amino acid molecules. Amino acids are the basic units which form proteins. එතකොට මේවා combine වෙලා එක එක જાති proteins හැදෙනවා. Different types of proteins are formed by the combination of amino acids, polymerization. So 20 amino acids have mainly contributed for the formation of proteins. So there are plant-based proteins, animal-based proteins, right? So there are different types of proteins. There are 20 such proteins. Test kala. So here it says, do you know plants are able to make all amino acids they need, but animals are unable to synthesize all the amino acids they need. Therefore, they must obtain some ready-made amino acids directly from food. These are termed as essential amino acids. So some in animal bodies, all the amino acids cannot be synthesized. So we have to take them as food externally. Right. So this is the structure, typical amino acid molecule, structure of a typical amino acid. Typical again, it is the model, right? There is a central carbon atom. There is a central carbon atom and there is a carboxyl group like this. A COOH කියන group එකට අපි කියනවා carboxyl group කියලා. ඊළඟට there is the amino group which is known as NH2 amino group. මෙහෙම NH2 කියලා ලිව්වට carbon combines with nitrogen. Nitrogen එක තමයි combine වෙන්නේ. Hydrogen දෙන්න දෙපැත්තෙන් ඉන්නවා. Then there is a hydrogen atom like this. And this is the variable group. May 20ක් තියෙනවා කියවනේ. There are 20 different amino acids. So this varies. This R group varies. For example, the simplest amino acid is glycine. In glycine, this R is equal to hydrogen. Hydrogen here also hydrogen, here also hydrogen in glycine. In another amino acid, here this R may be CH3. This R group changes and form 20 different amino acids. Another R represents a group containing both carbon and hydrogen. Due to the change in R group, due to 20 different amino acids are present. Extra knowledge jage habai make mataka tiyagan. There is a carboxyl group, amino group, hydrogen atom and the R group which varies. All proteins present in organisms from bacteria to human are made up of these 20 different amino acids combined in different sequences. මම කිව්වා අර අකුරු 20ක් අරගෙන ලියලා බලන්න ලියන්න යන්න එපා ඒක වෙලා යන වැඩක් 20 එක එක sequences වලින් ලියලා බැලුවොත් අන්න ඒ වගේ තමයි 20 different amino acids arranged in different sequences and form large number of proteins. හරි නේද? The simplest amino acid is glycine. Here hydrogen is present as the R group. එතකොට R වෙනුවට hydrogen තියෙනවා. අඳින්න ඕනේ නැහැ. හරි. These amino acids combine in different sequences to form අපි මේවට කියනවා form different polypeptides කියලා. Right? Polypeptide chains. Polypeptide on amino acids එක එක ඒවා combine වෙලා. If we just represent one amino acid, another one, another amino acid, then another one. Likewise, number of amino acids combine with each other to form polypeptide chains. මෙහෙම දෙපැත්තෙනුත් තව අයව අල්ලගෙන තමයි ඉන්නේ. Polypeptide chain කියලා කියනවා මේකට. Right? Polypeptides. Poly කියන්නේ many නේ. Polypeptides. Right? These are joined together by a type of bond known as the peptide bond, right? මේවා අතර තියෙන්නේ මෙන්න මේ bond එකට කියනවා peptide bond කියලා. peptide bond. Polypeptides are chains of many amino acids. A protein may possess one or more peptide chains. In a single protein, there may be one or more this type of polypeptide chains. Or when you do A-level biology, you will be taught that there is a primary structure, secondary, tertiary structure for proteins. කියලා දෙයි. දැනට මේ ඇති. So what are the protein rich food? Can Balan, I will write down when you say. Yes, protein rich food. Yes? අනේ කියන්නකෝ. Them. Yes, yes. Meat. Egg ගත්තොත් egg white. Egg white mainly consists of protein, right? Egg yolk contains lipids. Egg white portion of an egg. හරි. තව මොනවද කිව්වේ? Fish නේද? Okay, fish. Even dried fish. හරි. Then, yes. මොනවද පුතේ? Cereals and pulses. ඊළඟට දැන් අපි cereals and pulses ගන්න පුළුවන්. ඊළඟට grains. ඊළඟට mushrooms. Mushrooms become a supplement of proteins, especially for the vegetarians. May vegetarians do not consume any of these නේද? ඊළඟට soybean, right, beans. ඊළඟට අර සෝයා වලින් එක්ස්ට්‍රැක්ට් කරගන්න එකක් තමයි tofu කියලා කියන්නේ නේද? tofu soy milk product එකක්. tofu that is also rich in proteins. ඔයගොල්ලෝ සමහර විට ආස නැතුව ඇති. consume but it is rich in proteins. There is a different taste, but when you boil it for some time with salt, you will lose that unpleasant taste is lost then. So before cooking next time, you'd better to boil it in salt water for about 5 minutes. Hari, tofu. Tamanada, meat, fish, cheese, as you mentioned about pulses and cereals, lentils. Lentils කිව්වහම මොනවාද beans, long beans, winged beans ඒ ඔක්කොම ඇති. winged beans එහෙම ගොඩක්. මේ beans කිව්වහම bonchi ඊළඟට long beans, winged beans, all these are included. These are rich in proteins. Lentils, yogurt, nuts and seeds. Yes, even milk, mushroom. You can write those as well for protein rich food. But you have to consume the vegetables like beans, long beans, winged beans. They contain a lot of proteins. Done. හරිනේ? State the importance of proteins in living bodies. Let's see the importance of proteins in living bodies. So these are proteins are also used as an energy source when lipid and carbohydrates are not enough. Proteins also act as an energy source, right? Then it acts as a structural component when we consider the cell membrane. The cell membrane or the plasma membrane, there is a bit difference. In both plant cells and animal cells, this is available. So in plasma membrane, we find large protein molecules known as glycoproteins, right? Glycoprotein. As a structural component, these proteins become important. ඊළඟට දැන් අපි ගත්තොත් ඉන්නවා hair, nails, ඊළඟට feathers in birds අපිට නම් feathers හරිද? එතකොට they contain keratin. Keratin is a protein. ඊළඟට now when people become old, the skin becomes wrinkled. හැම රැලි වැටෙනවා නේද? That is due to the loss of elasticity in collagen fiber. Now, when you are young, like you all, when in small children and also in young people, because of the availability of collagen fiber in the connective tissue, so skin is also under the skin. Actually, we find connective tissues. So in those we find the collagen protein, right? Collagen. එකකින් එක ලියමුද ලියන ගමන් කියමු නේද? දැන් I mentioned 2 points. ලියන්නකෝ number 1. දැන් ලියන්න නැතුවනේ හිටියේ දුකෙන්. දැන් ලියන්න. Number 1, as an energy source. As an energy source. Number 1, as an energy source. අන්න අර කලින් වගේ කලින් ලීව් වෙන්න බෑ. අර කට්ටියක් ඉන්නවනේ. ගිය සැරේ 8 වෙනකොට disappear වෙන set එකක් හිටියා. දැන් එහෙම බෑ. You have to wait until the last moment. Now you are in grade 10, right? Hari, as an energy source. Under that you can say when the energy supply from carbohydrates and lipids is not enough, not adequate. Hari. Then number 2, as structural components. As structural components. So we'll give examples like this. එකක් තමයි glycoproteins in cell membrane in both plant and animal cells, right? Cell membrane. ඊළඟට collagen in connective tissues of animals. Animal connective tissues කියලා දාමු. එතකොට ලේසියිනේ. Animal connective tissues. Then keratin is a protein. Keratin in hair, nails, feathers, glycoprotein, collagen, keratin. Structures are made. ඒකයි structural components කියලා කියන්නේ. අනේ දැන් මූණට අත තියාගෙන දුකෙන් ඉන්න අත් පහල දාන්නකො දුක් වෙන්න එපා. Yes. හොඳට කොන්ද කෙලින් තියාගෙන ලස්සනට වාඩි වෙලා ඉන්න ඉතලාවත් the rest of the half an hour. Yes, Sakitya. Sansita. pain. Manusha, video on වෙන්න ඕනේ. ඉනුලි මොකද? ඉනුලි? Yashan, I didn't see. Oh, Savindu. Did I mention you about the checklist that you have to make when you are studying? කියලා දුන්නා නේ මං. I will try to එකත් හදලම දෙන්න බලන්නම්. දැන් තාම textbooks හම්බෙලා නෑනේ. Hands up those who got the textbooks. One, two. Only 3 of you. 4, 5, 6. Only 6. Hari, I will tell it once you get the textbook, right? You have to make a checklist to study. මේ කියන විදිහටම පුතේ කරන්න. මුල ඉඳන්ම මේ දැන් grade 10 පටන් ගන්නකොට මේ විදිහට කරගෙන යන්න, then it would be very easy. එතකොට හෙට term test එකට තියෙන වෙන subject එකක් අද නම් class යන්න බෑ. එහෙම ඒවා වෙන්නේ නැහැ. හරිද? ඔන්න දැන් හෙට grade 11 ඒ කියන්නේ grade 11 කියන්නේ O level batch එක. දැන් ඕගොල්ලෝ 10 නේ. එතකොට grade 11 කියන්නේ දැන් මේ අවුරුද්දේ 10 හිටපු අයනේ. ඒගොල්ලොත් නෙමෙයි O level set එක. ඒගොල්ලන්ගේ class එක හෙට තියෙනවා. ඔන්න දැන් message එනවා. හෙට term test මේ දවස්වලට ඔන්න අද term test පටන් ගත්තා. ඉතින් වෙන subject එකක් Thursday තියෙන්නේ. ඒ නිසා join වෙන්න බෑ කියලා. එම වෙන්න බෑ. ඕගොල්ලෝ මුල ඉඳන්ම හරියට වැඩ ටික කරගෙන යන්න. එතකොට හෙට test කිව්වොත් අද ගෙදරින් trip එකක් යන්න කතා කළත් යන්න පුළුවන් තත්ත්වේ ඉන්න ඕනේ. System you have to keep in touch, right? අම්මෝ එක්සෑම් එකක් එනවා ආයි ඔළුව හොල්ලන්නේ නැහැ කවුරුත් එක්ක කතා කරන්නේ ගෙදර නෑ දැයක් ආවත් එළියට එන්නේ වෙන්නේ නැතුව මුලින් ඉඳන්ම ලස්සනට කරගෙන යන්න ටෙස්ට් තියෙන දවස්වල විතරක් මහා එෆර්ට් එකක් දාන්න එපා හැමදාම හරියට වැඩ ටික කරන්න හැමදාම බැලන්ස් කරගන්න right පාඩනුත් කරනවා අනිත් ගෙදර වැඩත් කරනවා ස්පෝර්ට්ස් උත් කරනවා ඉස්කෝලේ අනිත් clubs and societies වලත් ඉන්නවා. ඒ ඔක්කොම ටික ලස්සනට බැලන්ස් කරගෙන යන්න. ඒකට ඔයගොල්ලෝ හොඳ ටයිම් ටේබල් එක අනේක දැන් පුළුවන්. චෙක්ලිස්ට් එක තව පස්සේ කියන්නම්. You can make the timetable now. දැන් දන්නවනේ ඊළඟ අවුරුද්දට මොනවද තියෙන classes. මොනවද what are the days in which I have to stay for sports or extracurricular activities. Then I will come home around this time. එන්නේ වගේ එක කාටවත් හදලා දෙන්න බෑ. ඔයගොල්ලෝම තමයි දන්නේ. You can take an A4 sheet, divide it into the 7 days, 7 columns හදාගත්තා. ඊට පස්සේ on Monday after school නේ කරන්න වෙන්නේ. After school මෙන්න මේ මේවට ගියාට පස්සේ ඔන්න I have to study this particular subject. Study කියන්නේ අර මා වෙලාවක් වාඩි වෙලා නෙමෙයි at least 45 minutes you have to allocate for one subject. In addition to the classes. class එකට ගියා කියන්නේ හරි පාඩම් කළා වෙන්නේ නැහැ. You have to allocate time to do the work alone. අද දැන් අද මේ ටික අදම බලාගෙන නිදා ගන්නවා නම් තමයි හොඳ. එතකොට හොඳට මතක හිටිනවා අර බලපු වීඩියෝස් ටිකත් එක්ක හොඳට මතකෙට යනවා. දැන් ওই ටික මම grade 6 ඉඳන්ම කියනවා. එහෙමත් කරලා නැත්තම් දැන් පටන් ගන්න. දැන් නම් අනිවාර්යෙන් පටන් ගන්න ඕනේ. මොකද you can't study 35 lessons within 1 week at least. I'm talking about science only. You have 8 other subjects. නේද? නිසා you have to have a balance. මේ ගෙදර එනවා දැන් මේ වෙලාව හරි I have to take dinner with my family members. අනිත් අය එක්ක කතා කර කර කෑම කන්න එකටත් වෙලාවක් තියෙනවා. කම්පියුටර් එක හරි මගේ වොයිස් එක පොඩ්ඩක් මේ දවස්වල අවුල්. කම්පියුටර් එක ඉස්සරහා හරි ටීවී එක ඉස්සරහා හරි එහෙම නැතුව කට්ටිය එක කතා දවසේ වෙච්ච දේවල් ෂෙයා කරගන්න ගමන් එහෙමත් වෙලාවක් වෙන් කරගන්න හැමදාම ඒක තියෙනවා. ඊට පස්සේ I before I go to bed I have to do the homework. school homework කත් කරලා ඒක විනාඩි 45ක් නැත්නම් පැය භාගයක් හරි you have to allocate for one subject to revise the previous thing. ඊළඟට I told you don't go for number of classes you attend is not directly proportional to the result. Science නම් මං කියන්නේ මං ළඟට මෙන්න කියලා මොකක් හරි ඕගොල්ලෝ සිලෙක්ට් කරගන්න එක ක්ලාස් එකට එකකට ගියාම ඇති right සයන්ස් ක්ලාසස් ගොඩකට ගියා කියන්නේ සයන්ස් වලට හොඳ රිසල්ට් එකක් ලැබෙනවා කියලා එහෙම එකක් නැහැ right you have to get the things from school and extra support from the classes then you have to do the work alone so I guaranteed on the first day itself if you do the work according to the advices that I give you you can go for a super A pass. ඔව්. ඒක ආයි ප්‍රශ්නයක්ම නෑ. ඕන තරම් අපි ප්‍රශ්න කතා කරලා ඉවර කරනවා. ඔන්න මම ඔයගලන්ට ඕනම් පෙන්නන්නම් පාස් පේපර්ස් ටික ඔක්කොම ඉවර කරනවා. දැන් මම මෙහෙම chart එකක් හදාගෙන 2024 O level ඉවර වෙලා results උත් ආපු batch එක. ඔන්න ඒගොල්ලන්ට පාස් පේපර් ටික කරපු විදිය. හරි 2016 onward. ඊට පස්සේ දැන් මේ හෙට අනිද්දා යන්න ඉන්න සෙට් එකට for February මේ ඒගොල්ලන්ට ඔන්න questions ඉවර කරපු විදිය pages දෙකක තියෙනවා යාළගේ right this one ඔන්න ප්‍රශ්න ඔක්කොම ටික කතා කරලා අවුරුද්දෙන් අවුරුද්ද වැඩි වෙනවා ඊට පස්සේ දැන් grade 11 ඉන්න අය දැන් grade 11 වලට යන අය 2026 O level ඒගොල්ලන්ටත් දැන් ප්‍රශ්න පටන් අරන් තියෙන්නේ මේ විදිහට ඔයගොල්ලන්ගේ ටිකක් කල් යන්න ඕනේ O level papers වලට යන්න ඉතින් ඒ වගේ පිළිවෙලකට පේපර් ක්ලාස් එකේ ප්‍රශ්න ටික කතා කරලා තියරි එකේ හොඳටම තියරි කියලා දීලා then some second lesson for example we have lot of calculations to do. එතකොට in the theory class also we are doing lot of calculations. ඊට අමතරව තමයි paper class එකේදී we are discussing the questions that came for the exams. right? ඉතින් ඔය ටික කතා කළාම ඔයගොල්ලෝ ආයි තව තැනක හිට ගන්න යන්න ඕනේ නැහැ. Definitely you can go for a super A pass. හරිද? ඒ කියන වැඩ ටික කරන්න ඕනේ මම කියන්නේ මම හිතන ක්ලාස් එකේ ඇවිල්ලා වාඩි වෙලා හිටියා කියලා සුපර් ඒ පාස් එකක් හම්බෙන්නේ නැහැ you have to follow the instruction ඒ නිසා අනිත් සබ්ජෙක්ට්ස් වලටත් එහෙම ඔයගොල්ලන්ට සපෝට් එකක් ඕනේ ඒවට හැම සබ්ජෙක්ට්ස් එකටම හැම සබ්ජෙක්ට් එකටම ක්ලාසස් වලට යන්න ඕනි කියලා එකකුත් නැහැනේ සමහර අය මොනවටවත් යන්නේ නැතුව කරන අයත් ඉන්නවා right ඉතින් ඒ විදිහට ඔයගොල්ලෝ දන්නවනේ මට I need some extra support for this subject යාළුවා කරන නිසාවත් අරයා යනවා මෙයා යනවා ඒව ඕනේ නැහැ you think of yourself right ඒ විදිහට you can make a timetable එතකොට ඒ හැම දවසෙම allocate කරන්න පොඩි දැන් classes නැති දවසුත් ඇතිනේ වැඩි වෙලාවක් ගෙදර ඉන්න දවස් ඇති හැබැයි definitely you have to allocate time for the other things as well to engage in your religious activities ඒවත් කරන්න ඕනේ right if you can allocate a small time for uh, to do a mindfulness meditation ඒ වගේ දෙයක් ඒක mindfulness is not something only for Buddhists right in other countries also people are practicing then your mind will be relaxed can you will learn how to concentrate on something at the mindfulness techniques මෙතන කියලා දෙන්න යන්නේ නැහැ මම if you search in the internet YouTube you can find lot of methods ඒවා පොඩි දේවල් මහා දේවල් නෙමෙයි you can just scan your body close your eyes and just body scanning කියලා කියන්නේ මේ අපි ඉන්න දිහා බලාගෙන කොහොමද මොනවද දැනෙන්නේ ඇඟ දිගේ අර මනස අරගෙන එන එකක් ඊට පස්සේ you can count from 100 to 100 to 1 meditation කියලා තියෙන ඒ වගේ පුංචි දෙයක් හැම දවසෙම කරලා පාඩම් කරන්න පටන් ගන්නවා නම් that would be very good because then your අපි කියන්නේ අර you are trying to reach the alpha state එතකොට you can remember the things very well. එතට මතක හිටිනවා අර වෙන වෙන දැන් වාඩි වෙලා පාඩම් කරන ගමන් හිත ගෙදර ඉඳන් පාඩම් කරනකොට හිත ඉස්කෝලේ තියෙන්නත් පුළුවන්. එතකොට you are not concentrating enough. When you are practicing a mindfulness technique like that, you can concentrate. එතකොට efficiency of your uh, sacrification or the commitment that you are making will be high. right because you are concentrating එතකොට අර එකම දේ ගොඩ සැරයක් පාඩම් කරන්න වෙන්නෙත් නැහැ හොඳට මතක තියාගන්න පුළුවන් වෙනවා ඒ වගේ අදම නෙමෙයි දැන් අද අමාරු නම් හෙට හරි ෆ්‍රී ටයිම් එකක් හොයාගෙන දැන් ආයි නිවාඩු වෙනවනේ දැන් from 23rd onwards නේද? ඉතින් එතකොට ඒ වගේ පැත්තකට වෙලා වාඩි වෙලා ටයිම් ටේබල් එකක් හදාගන්න. ඒක හදාගෙන බිත්තිය අලවලා වෙන වැඩ කරන්න නෙමෙයි. ඒක ඒ විදිහට වැඩ කරන්න try කරන්න. ඒක වෙනස් වෙන දවස් එන්න පුළුවන්. මොනවා හරි විශේෂ හේතුවක් නිසා it's okay. But try to follow that timetable as much as possible. Saturday, Sunday, එතකොට අර religious activities, some time to spend with the family members. For your hobby, you have to have a hobby, right? එහෙම එකකුත් තියෙන්න ඕනේ. නේද? ඒක hobbies තියනවානේ. එහෙම එකකුත් තියාගන්න. You can just enjoy. ඒ කියන්නේ hobby කියන්නේ just staring at the uh, computer or the television නෙවෙයි, right? Something that you are engaging actively. එම දෙයක්කටත් පොඩි වෙලාවක් දාගන්න. හැමදේම balance කරගෙන subjects, all the 9 subjects are also covered within the week in addition to the classes. Right now Tuesday science class නම් අද පුළුවන් නම් හොඳයි. අද බැරි වුණත් at least before you go to bed tomorrow, because the things will retain in your mind for at least 24 hours. එතකොට අදම පුළුවන් නම් හොඳයි. නැත්නම් කොයි වෙලාවටද මම පාඩම් කරන්නේ? රැයටද පාන්දර නැගිටින්න පුළුවන්ද? එහෙමත් සමහර අය රැයට හැරිලා ඉන්න පුළුවන්. සමහර අයට පාන්දර හැරි. You have to have a good sleep as well. කොට ඒ ඔක්කොම ටික හදාගන්න. Just plan the things. හරිද? එතකොට මේ grade 10 මුල ඉඳන් ඒ විදිහට ගියාම it would be very easy for you to do the stuff, manage the stuff. When the exam is close by, you are not under stress. Hari, okay, I can go and sit for the exam without any problem. You are ready. හරිද? එහෙම හදාගන්න මුලදිම. Okay. හරි. දුර ගමනක් ගියා. Number 3. ඒ මොකද්ද කරේ? ලියලා ඉවරයිනේ. Number 3. The third importance of proteins. You can say as enzymes. As enzymes. හරි. මොනවද මේ enzymes කියන්නේ? Now in the laboratory, if we are doing a particular experiment, if we need to do a particular chemical reaction, sometimes we have to do certain things in order to speed up the reaction. Sometimes we will have to heat it up. Sometimes we have to add a catalyst. On the catalysts we are going to discuss in coming lesson. So catalysts are the substances which will speed up the rate of a biochemical reaction, right? The substances which speed up the rate of a biochemical reaction. Those are known as catalysts. So enzymes are, there are so many biochemical reactions happening inside our body, right? So many biochemical reactions happening inside our body. So we can't do something externally for them. These are naturally speeded up by a biochemical catalyst. That is, those are known as enzymes. Enzymes are known as biochemical catalysts. දැන් අපි biological catalysts කියලා කියනවා. These are available in living bodies. So all the enzymes are proteins, right? All the enzymes are proteins. They speed up the biochemical reactions happen inside the living bodies. අපි catalysts ගැන මේ පාඩමේම කියනවා about the enzymes. ඊට පස්සේ catalysts ගැන වෙනමත් කතා කරනවා in grade 10 third term. හරි. එහෙනම් එතෙන්ට ලියන්න under the third point you can say all enzymes are proteins. All enzymes are proteins. Underline all. All enzymes are protein. Then you can say the biochemical reactions. Biochemical reactions. Biochemical reactions biochemical reactions that take place in living bodies, that take place in organisms, that take place in organisms that take place in organisms are catalyzed by enzymes, are catalyzed by enzymes. Are. Number four, fourth one, uh, as hormones, as hormones. Not all hormones, on all enzymes are proteins. Some hormones are having a protein origin. So the hormones, for example, if we take insulin, growth hormone, so insulin is responsible for controlling our blood glucose level. So it is coming under homeostasis. Homeostasis. There is a separate topic once again in future. Homeostasis. Homeostasis. That is the maintenance of our uh, the conditions in the internal environment of the body at an optimum level. For example, blood glucose level should be maintained, body temperature should be maintained. So the blood glucose level is maintained by insulin. Then growth of the body is controlled by the growth hormone or GH. Right? These are having a protein origin. Some of the hormones are having a lipid origin. Right? Growth hormone, GH. Lipid origin තියෙන hormones ගැන අපි ඊළඟට කියමු lipids ගැන කියද්දි. Right? The coordination part is done by the hormones inside the living bodies. Now, as hormones කියලා ලියලා, under that you can say some hormones have a protein origin. Some hormones have a protein origin. Examples you can say insulin and growth hormone. Insulin and growth hormone. Then you can mention they involve in homeostasis. They involve in homeostasis and coordination of organisms. Coordination of organisms. Yes. They involve in, is that the sentence you want me to repeat? They involve in homeostasis. They involve in homeostasis and coordination of organisms. And coordination of organisms. Yes. I mentioned that some hormones have a protein origin. Some hormones have a protein origin. Examples, insulin and growth hormone. Then I told uh, they involve in homeostasis and coordination of organisms. Homeostasis and coordination of organisms. හරි නේද? Right. Then number 5 as antibodies. Antibodies are responsible for, actually these are produced by the white blood cells. These also have a protein origin. They protect our body from pathogens, right? Pathogens are the disease causing agents. And as antibodies, under that you can say uh, these are produced in the body. These are produced in the body to protect our body from pathogens. To protect our body from pathogens. Entering the body. Entering the body. Uh, these are produced in the body. These are produced in the body to protect our body from. Are produced in the body to protect our body from pathogens. And as the last one, we can say as respiratory pigments. As respiratory pigments. Now in our body, in our bloodstream, oxygen is transported through red pigment, hemoglobin. Hemoglobin, right? Hemoglobin. So in hemoglobin, heme is iron and globin is the protein component. Heme is iron, globin is a protein. Now you can say as respiratory pigments and then write the example hemoglobin. හරි protein test එක කියලා ඉවර කරමු. එතකොට proteins උත් ඉවරයි. Then next week we can start lipids. We can discuss about lipids and nucleic acids. Next week වෙද්දී අපි second unit එකත් පටන් ගන්න බලමු. If I share the tube, you take a printout and join the next session, right? Hari. In addition to this, remember, no need to write. The venomous substances are also the toxins, also mainly contain proteins, right? Snake venom wage. Hari. Then finally, describe a test to be done in order to identify proteins. There is a simple test which we call as the biurate test. සමහර අය මේක biurate test කියලාත් pronounce කරනවා. Actually that is the biurate test, right? There what we can do is we can obtain a protein extract. Then we mentioned egg white contains proteins කියලා. ඒ වගේ you can take some egg white into a test tube, or you can grind dal and mix it with water and obtain the extract, right? So you have to have a solution containing protein. Then යමු ස්ටෙප්ස් ටික. හරි. So you have obtained a solution containing proteins. ඊට පස්සේ we can add මෙතන biurate reagent කියලා එකක් පාවිච්චි කරනවට වඩා we can to follow these two steps. You have to add an extra amount of extra volume of sodium hydroxide and few drops of copper sulfate solution. හරි. Add number 1 volume of sodium hydroxide NaOH. Now we will learn how to write the chemical formula in the third unit. Sodium hydroxide NaOH. Then few drops of copper sulfate solution. Few drops of copper sulfate solution. That is CuSO4, copper sulfate solution. Then here you are shown this is a negative result and this is a positive result. Positive result means the observation is you will be able to obtain a pinkish purple color. Right? Observation. Negative result can this has not contained proteins. හරි. ඕක ලියාගෙන ඊට පස්සේ we will watch the video and stop for today. Hope you will study about both carbohydrate protein day itself or tomorrow in the early morning. නේද? Yes, have you finished writing? ඉවරද? සයිල් හරිනේ? සංසිත Ernestine, did you get late to join or what happened? Did you get disconnected in between? Okay. So if you have missed a lot, you can watch the recording, right? I will share it by tomorrow. Hari, shall we watch the video then? You all have finished writing. Arine, and I'm going to share the video. So here you can see sodium hydroxide. It does not have a color. Copper sulfate solution, there is a light blue color. You know the color of copper sulfate. And here they have taken an egg. So we can obtain the egg white.
Correct. Then what?
So it should be that multigrain bread, right? Normal bread that we eat will not contain that much protein. You consume multigrain bread, then it will work. Actually, when we are mixing the things, we should not use our thumb, right? These things should not come in contact with our skin. They can, these may not be harmful, but in the case, in this case of some of other chemical substances, We should not mix it like that. And the other thing is the things on our hand will go into the mixture. Because of both of these reasons, we should not use the thumb like this, right? That is a wrong example. So you can see egg white gave the purple color here. Milk also has given, but salt, there is a negative result. Salt negative result. Egg white positive, milk is also positive. That is why we mentioned pinkish purple, right? හරි නේද? එහෙනම් අපි අද කතා කළා we discussed about the locations and the important facts about carbohydrates, the three types, and then we discussed about proteins. Next time we are going to discuss about enzymes and then lipids uh, and then about nucleic acid, right? කතා කරගෙන යමු අපි sometimes I will share the tute for the second unit. දැන් මම ටික දවසක් බැලුවා because of the affected students because of the bad weather some were affected ඉතින් එතකොට the second unit කියන්නේ something very important. එතකොට ඒක මිස් වෙන එක පව් නිසා දැන් මං හිතන්නේ කට්ටිය stable වෙලා තියෙන්නේ නේද? They have become stabilized. Therefore, sometimes I will share the unit tute for the second unit. So if I share this before the next session, that is on the 23rd, right? You can take a print out and join. හරි අපි 30th නම් ඔන්න හොලිඩේ එකක් දීලා තියෙනවා since it is year end නැත්නම් දවස් 5ක් තිබුණාම එකක් නිවාඩු දෙන සිස්ටම් එකක් නැහැ මගේ අර year end එක නිසා some have celebrated Christmas ඉතින් අපි අවුරුද්දක් මහන්සි වැඩ කරලා අන්තිම සතිය ඔන්න නිවාඩුවක් දීලා තියෙනවා the 30th but we are having the session on the 23rd right හරි ඉන්න බලන්න ඒකත් මතක් කරලා ඉන්න ෂෙඩියුල් ඔයාලගේ කලින් ඔයාලට ක්ලාස් එකක් දාලා තියෙනවා ඒකත් මතක් කරලා දීලාම යවන්න ඕනේ yes 23rd Tuesday we are having the session and 24th Wednesday also early in the morning. එතකොට 23 හවසත් හම්බෙලා 24 උදේමත් හම්බෙන්න තියෙනවා. අපිට ගිය සතියේ මිස් වෙච්ච session එකට on the 24th I have put a session. මට කියන්න චුට්ටක් the Catholic or Christians you don't go to the church on the 24th early morning, right? නෑ නේද? 24th early morning යන්නේ නැහැ. I have put the session from 7 to 9:30 in the morning, right? ඒකත් මතක තියාගන්න 24th morning. හරි. එහෙනම් let's wind up the session for today. Let's meet on the coming Tuesday, right? Hari. Stay safe and have a nice day. Nice time and please study the things. Okay? Good night.